What is a storage disease? We're going to start in pedestrian terms. So you start out with a complex substrate. This is an extracellular molecule. Cells can take this up and undergo the normal lysosomal degradation pathway into small little products. Those little products can be used inside the cell or excreted. So, a storage disease is when you have these products are taken up by cells and they are not processed properly. And there's different types of storage diseases. We'll go through the several different ones. What that means is that you have an unusual accumulation of molecules that shouldn't be there. So this principle is the same thing that we've seen with any biochemical pathway. If you've got material moving through a pathway, if you have a block on the pathway, you better have an escape route for the upstream substrates. So for example, blocking HMG-CoA reductase blocks the conversion of HMG-CoA, but we can reverse that back to acetyl-CoA and we can put that into fatty acid synthesis. So we're not building up HMG-CoA to toxic levels. This is a violation of that principle. If we have a block, we're going to build up this substrate to toxic levels. Okay? So that's the principle of pretty much all storage diseases. Now, the first one we need to talk about are glycogen storage diseases. So glycogen storage diseases, you might think that they're typically either you cannot store glycogen, let's say deficiency in glycogen synthase, or it's a deficiency in glycogenolysis, so a deficiency in glycogen phosphorylase. Remember I said that enzyme was going to be real important to you. So in muscle, under normal conditions with liver, the liver is putting out glucose. Cells can take up this glucose and use it for oxidation. Muscle cells will take up the glucose under the fed state, make glycogen, glycolysis, make energy, normal state. Now what happens with a glycogen storage disease? What we are primarily talking about is a defect in glycogen phosphorylase. So what does that automatically tell you about what's going to happen? So this is defective. What do you propose seeing, let's say under histochemistry? So you take a biopsy of skeletal muscle, you stain it with H&E. What do you expect to see with a glycogen phosphorylase deficiency?
Lots of glycogen.
Lots of glycogen, yeah. Okay, so I know this is kind of simple, but I'm going to do this right this year. So lots of glycogen. So that tells you that the glycogen synthase complex is perfectly fine. The normal mechanism can work and we can store glycogen. We just can't access that. So what kind of symptoms would a patient feel?
Low energy.
Cramps, myalgia, low energy, muscle stiffness.
Everything the last guy had.
Everything the last guy has, right, rhabdo. So you're going to lose your capacity for short-term energy needs. Now, does that mean the muscle is shut down and you'll just never actually move your muscle? No. Because what do you can rely on?
Eat more often.
Eat more often, eat like a bird, get that GLUT4 going.
Or fat, yeah.
Or fat, right. So these symptoms, glycogen storage diseases in muscle, tend to be rather clinically mild. Because a person's not going to have energy, they're going to be fatigued. We're going to see that in our case after this one here. It's called McArdle's disease. They're not going to have a lot of energy, but they can solve the problem with fat intake. What about the liver? A glycogen phosphorylation mutation deficiency in the liver. What do you think that patient will present with?
Hepatomegaly.
Hepatomegaly, big time, yeah. What are they not going to be able to do with a deficiency in glycogen phosphorylase? Maintain their glucose, serum glucose, yeah. Low blood glucose. So these people are hypoglycemic. Now, do you think this is a mild or severe?
Severe.
Severe, yeah. Simply because the liver's job is to keep that plasma glucose up. Because if that plasma glucose goes down, your brain starts to become
Okay.
That's what happens when your brain decompensates. No, that's the liver. So that's serious. Because the liver is supposed to supply the glucose for the brain, and it does so by glycogen and
Ketones.
supplying glucose. I'm getting ahead of myself, but I'm pinging your biochemistry for the next couple of days.
Gluconeogenesis.
Gluconeogenesis, yeah. So in an individual like this, is gluconeogenesis going to be impaired? No, they're perfectly fine. Because the glycogen phosphorylase enzyme is a side reaction to the gluconeogenic pathway. So they'll be able to maintain some glucose levels. But gluconeogenesis is insufficient to keep up. And that's why ultimately we're going to talk about going into ketones, because ketones will kind of pick up the slack. Okay? So now here's some different enzyme deficiencies. So here's the two that we just talked about. McArdle's disease. These are mild symptoms, muscle glycogen phosphorylase. You can see how rare these all are. Liver, glycogen phosphorylase. Mild, severe, mild. Is that mild or severe?
You just said it was severe.
Yeah, it's pretty severe, the hypoglycemia. I don't know why I wrote mild. Down. Because the glycogen phosphorylase can work up to a branch point but not beyond it. So that's why the branchers. So would these be mild or severe symptoms? So skeletal muscle, can it obtain glucose from its glycogen with this mutation?
Yeah.
Sure.
Mild.
How about liver? Can it resupply the plasma with glucose?
Not well.
Not as well.
Compromised, but it still can. So these will be mild.
Wouldn't you end up with like large accumulations of glycogen in the liver as well? Because if it can't be branched, then you just keep building it up that way.
So the question is, wouldn't you end up with a heavily glycogen storage? So you're going to increase your glycogen storage capacity, you're right. But that's going to be dependent on a balance of the glycogen synthase activity and the branching enzyme on there, right? But you're right, ultimately you're going to start increasing the amount of glycogen storage. What's this one? Acid maltase, Pompei's disease. Acid maltase, alpha-glucosidase, lysosomal deficiency. What's that got to do with a glycogen problem? This is an alternative path to generate glucose from glycogen. So the normal glycogenolysis occurs in the cytoplasm. But there's a mechanism that is completely uncharacterized that some of that cytoplasmic glycogen can get into lysosomal compartments. And once you're in a lysosomal compartment, you've got to cleave off that glucose residues. There is no glycogen phosphorylase in the lysosome. So we use acid maltase because of the low pH in the lysosome, and we use the enzyme called maltase. Okay, that leads to Pompei's disease. So now, what part of this do you need to know for the block? Well, these different diseases, you need to know McArdle's disease because this specifically is the case we're going to be looking at today. But the names of all of these other ones, you need to know those for the step one, but not for this block. What you need to know is that if I give you a mutation in one of these different enzymes, you should be able to predict what the clinical phenotype is going to present. Okay? And that tells me that you understand the mechanism of glycogen regulation. So there's another nifty summary chart for you. Want to keep going with this high yield information? Okay. So that's glycogen storage diseases. So let's now talk about lysosomal storage diseases. So the normal function of a lysosome, here's our picture. We've got endocytosis, early endosomes. We have trafficking of this material to the late endosome, and then that's going to traffic to the lysosome. The sole purpose of the lysosome is the garbage can. So its job is to degrade everything that goes into the lysosome needs to be degraded to its constitutive parts. So any kind of triglyceride that gets in there needs to go to fatty acids, proteins to amino acids, polysaccharides all the way down to the monosaccharide unit. Now, how does acidification work? We have a protein on the surface of the lysosome. It actually starts out in the late endosome, and it's called a VATPase. The V stands for vacuolar ATPase. This is the common proton pumping transporter that's involved in controlling pH in these lysosomes. So the job here is to take ATP, hydrolyze ATP, and use that to pump protons into the lumen of the lysosome. Now, we need ATP hydrolysis because the concentration of protons is going to dramatically increase as that pH goes down. So we're going to be pumping against a gradient, hence the ATP necessity. Now, why do we need to drop the pH? Because this is the one place inside the cell that we have a lot of dangerous enzymes. Proteases, lipases, glycosidases, DNases, and RNases. So we saw all of these being synthesized and secreted by the pancreatic acinar cells. Now the pancreas controls these activities by having cofactors, or in the case of the proteases, we need enterokinase activation. Then these proteases are activated in the proper environment in the lumen of the intestine. That lumen is pH 7. Okay, neutral pH. What would happen if the cell made all of these proteases and lipases, these hydrolases, and they were active in a pH of 7? Autodigestion. It would be the same thing that the pancreas occurs, but on a smaller scale. So these are separate enzymes from any of those enzymes we've seen before. And these are all called acid hydrolases, and they work at low pH. So at neutral pH, these enzymes are inactive. They do not have to become activated, so they are not zymogens. They're synthesized in their active structure, but they require a low pH environment to perform their catalytic activity. And hence, we keep them controlled in the lysosome. Okay? Now, one of the big lysosomal storage diseases is transport of these molecules. So these molecules are made just like any other secreted proteins are. They're made in the endoplasmic reticulum. So remember, we have our gene in the nucleus. We make our mRNA. The mRNA, we have our ribosomes binding to this. And so what I'm drawing is the same structure, the same path here that insulin takes, making our pre-proinsulin, our proinsulin, and full insulin, mature insulin. Once the ribosomes bind on this, then they can bind to the endoplasmic reticulum. That's what all these dots are. Those are ribosomes. And we're secreting the protein as it's being translated into the ER. Now, what's called the constitutive pathway is the secretory pathway. So anything that goes into the ER is going to translocate to the Golgi. It'll become modified there. And then it's going to go to the trans-Golgi network. And then it's going to go, typically, to the plasma membrane for secretion. And this is called the constitutive pathway. Constitutive means it's not regulated. Everything just goes in that naturally. Now, what if we want to send the proteases and lipases and hydrolases that we're making in here, what if we want to send them to the lysosome? We need a targeting sequence. And that targeting sequence is going to be a mannose 6-phosphate. So all of the enzymes that we're going to make that need to go into the lysosome are all going to be tagged with this mannose 6-phosphate. Now, when these enzymes percolate through the Golgi and they get to the trans-Golgi network, we are going to have a mannose 6-phosphate receptor. So all of these enzymes that have this mannose 6-phosphate tag, mannose is just a simple 6-carbon sugar with a phosphate on it. They're all going to bind to these receptors, and they're all going to be sequestered into a corner of this trans-Golgi network. This gets budded off, and then it gets delivered to lysosomes. Now, once this complex reaches the lysosome, it encounters the reduced pH because of that VATPase transporter. Then the hydrolase that's bound to the receptor dissociates because of that low pH. The hydrolase goes to the lysosome, and the receptor is recycled to the Golgi. So it's a very similar pattern as the LDL receptor recycling. You need to transport your cargo, which is the hydrolase, to the lysosome, and then you're going to recycle the receptor for continued rounds. Now, one of the biggest lysosomal diseases is called a mucolipidosis. And this particular one is called eye cell disease. And eye cell disease is simply a defect in the phosphotransferase that confers the mannose 6-phosphate signal. So this is the biochemical path on here, but the key evidence here is that this mannose 6-phosphate is no longer here in this deficiency. So this first enzyme, the UDP N-acetylglucosamine, this is not going to be working. It's not going to be active. Don't have to worry about that enzyme. The key element here is that the mannose 6-phosphate signal is not going to be on the hydrolase. So what does this particular mutation mean to your lysosome? Will the lysosome get the hydrolases? No. Where will the hydrolases go? Extracellular space, yeah. The sole problem with this is that you're not clustering because you don't have the signal, so you're not clustering these hydrolases in this transport vesicle and sending it to the lysosome. So the only way they can get into this transport vesicle is by diffusion. And so some actually get in there and are transported, but the concentration's too low. So most of them end up getting secreted in the constitutive pathway. So why is this called eye cell disease if you don't have hydrolases getting to the lysosome? Inclusion bodies, yeah. So what these look like under a microscope is you'll see a lot of lysosomes in there, but they'll have this pattern. So if this is a lysosome, it'll have this kind of whorl-like pattern to it. And that's called eye cell disease for inclusion bodies. And all this stuff is, in the lysosome, are the proteins, the lipids, the carbohydrates, possible DNA and RNA that's in there that has not gotten degraded. So this is probably, if I remember correctly, the first lysosomal storage disease that was ever discovered, eye cell disease. It's been around so long, I remember it when I was a student. Okay, another, mucolipidosis. So it's called a mucolipidosis because primarily those whorls in there are primarily lipids that you can actually see and they get stuck in there. So here's another one. Sphingolipids. So sphingolipids are the specialized lipids. We'll go into these a little bit more next week. But they're a category of complex lipids. So here's our phospholipid, our membrane lipids. If you add a fatty acid here, you'd have a triglyceride. So a sphingolipid has a little bit different structure to it, different components, but the backbone structure is the same as a triglyceride. So our sphingolipid here is this structure, and what's one of the most common sphingolipids that you know? Sphingomyelin, yeah, that's this one. And that's simply a choline residue. Here's your choline residue added on the phosphate group here. And this is on myelin sheaths. So that's the predominant fat on myelin sheaths. What's a common sphingoglycolipid? They're an antigen you use in blood typing. The blood groups, ABOs, that is a sphingoglycolipid. So when you're blood typing, looking for the ABO on an individual, what you're looking for is the pattern of this particular carbohydrate on a sphingoglycolipid. Yes?
Move the paper down just a tiny bit.
Oh, yes.
Thank you.
I'm not used to a paper up here. Okay, so now what happens on these? This is one. Who in here knows Dr. Garber? Oh, good, a few of you do. You know what he works on in his lab? Niemann-Pick disease. Here it is. Niemann-Pick. So Niemann-Pick is a brain development problem, primarily because this particular protein, which is called the Niemann-Pick, C1, NPC1, is deficient in these individuals. And so what happens in Niemann-Pick disease is that these individuals cannot process sphingolipids, primarily sphingomyelin. So cells will construct sphingomyelin and they'll also bind it and internalize it while they're doing their remodeling and construction, especially in neurodevelopment. In the lysosomes of these individuals, the enzyme that's absent is sphingomyelinase. And so if these individuals either have a deficiency in this enzyme or more commonly a deficiency in this protein that transports sphingomyelin, they will have this particular lipid disease called Niemann-Pick disease. It's a severe neurodegeneration disease, and I remember Dr. Garber telling me that children typically don't live much past 6 or 7. Although, does everybody know Dr. Heidenreich? Yeah. He with Dr. Garver together developed a modified treatment to actually extend the lives of these kids using cholesterol extraction. It's a complicated story, but it really works. That's their claim to fame. So I'm piggybacking on that research because it's really cool stuff. So what are some of these other lipid diseases? Fabry's disease, Gaucher's disease, Tay-Sachs. These are all lipid storage diseases. And the primary problem here is that there's a severe lack of these particular enzymes. And again, it's a deficiency in these enzymes, not the transport of the mannose-6-phosphate signal. That's only eye cell disease. But these particular diseases are deficient in these enzymes. And so these lipids corresponding to these diseases are built up and stored in those lysosomes, eventually causing toxicity. Okay, so that's the lipidosis. So again, the common theme is that you have an abnormal accumulation of these particular lipids, just like we saw an abnormal accumulation of glycogen. And each disease will have its enzyme that's deficient. Okay, the last one is not a mucolipidosis, but a mucopolysaccharidosis. So this is the last category of storage diseases. The principle is exactly the same as a glycogen or lipidosis. It's a storage problem, but in this case, it's a storage problem of the glycosaminoglycan proteoglycan structure. So what are these? Well, that matrix protein that's put down, carbohydrate that's put down in cirrhosis, we looked at as hyaluronic acid, hyaluronate. This is a major component in extracellular matrix, and it is absolutely the largest glycosaminoglycan. It can have up to, I think, 50 to 100,000 residues of carbohydrate. And each one of these monosaccharides is a hexose. And they can have 50 to 100,000 of these units. It's huge. Now we have other proteins that can associate with it that can have other carbohydrate branch chains coming off of the protein. And these can be keratin, chondroitin sulfate, heparan sulfate. And all these structures are, the difference between these carbohydrates, they're all hexoses. So they're modified glucose structures. And you'll have some here that are iguronic acid. You'll have N-acetylglucosamine. So all these are modifications of the glucose structure. Some will have sulfates, some will have acetyl groups on them, but they're all modifications with their own names, and these structures are part of the extracellular matrix. So you can imagine during development with remodeling structures, these are laid down for cell migration and cell attachment, and then during remodeling, they have to be taken up by cells, sent to lysosomes, and degraded. The two common ones are Hurler syndrome and Hunter syndrome. And these are deficiencies in the enzymes necessary for degrading these proteoglycan, glycosaminoglycan structures. So Hurler syndrome is iuronidase deficiency, and Hunter's is iuronidate sulfatase deficiencies. So one enzyme in Hunter syndrome takes the sulfate groups off, the other one actually hydrolyzes off the iguronic acid, which is a carbohydrate monomer. Yes?
Could you write out those enzymes a little bit bigger?
Sure, absolutely. This is alpha iduronidase, and this is iduronate.
Sulfate.
That's a misspell in that figure. It's iduronate. Yeah. So one enzyme cleaves the sulfate groups off. which are, a good example is right here. There's a sulfate group, there's another one. And then the other one will cleave off the iguronic acid. Iguronic acid, we do not have an example in this figure on here, but it's just a hexostructure. So again, deficiencies in these enzymes will lead to an accumulation of this structure in the lysosome and a storage disease. Okay, so what do you need to know from me for the block? Well, to me is just the definition of a storage disease. So if I give you an enzyme that would be mutated, say somebody presents with a biopsy of muscle that shows large amounts of glycogen, which of the following enzymes is more likely mutated? I will not ask you a question about Hurler syndrome or Hunter syndrome and ask you to give me the definition of that. That's a step one thing. That's one of those things that, you got to memorize the enzyme with the disease and then you can flush it after the exam. Or not, depending if you like it.
If you want to be a geneticist.
Yeah, unless, of course, yeah, you're right. These are all inborn errors, so that's where Dr. Heidenreich becomes your best buddy. Okay, questions on storage diseases. Okay, so we got glycogen. That's the most common one. We've got lipid storage diseases, of which there's two kinds. There's one that's eye cell disease that is actually a defect in transport of all hydrolytic enzymes to the lysosome. We have defects in lipid processing enzymes, such as sphingomyelinase in Niemann-Pick. And then we have our mucopolysaccharides, which are the glycosaminoglycans that get stuck in that lysosome because we're missing these particular processing enzymes.